All right. Hi, everybody. My name is Delaney McWalters. I'm a marketing consultant working with the Lupus Foundation of New England, and I'm very honored to be joined by Jillian Richmond here today out of UMass Medical School, a researcher in the field of lupus. Jillian, I'll let you introduce yourself.
Hi, everyone. I'm very happy to be here, and thanks to Delaney for interviewing me and for Beverly from Lupus Foundation for inviting me. And Trisha Santos, too. I almost forgot Trisha. So I am a scientist at UMass Chan in Worcester, Massachusetts. I am a T-cell biologist by training. So T-cells are a type of white blood cell that can actually attack your own body's tissues in the context of lupus, but they can help keep you safe from things like infectious diseases and cancers. So if any of you have a loved one or a friend who may have received cell therapy for cancer, that's actually a T-cell therapy that they use now. So we're starting to look into ways that you can use T-cells for therapeutics for lupus. And some of the new treatments, including the CAR T-cells that attack B cells, are very good at clearing the autoreactive or self-reactive B cells. It's a different type of white blood cell. So basically, we study the adaptive immune system and how the T cells and B cells interact and how we can try to harness that to treat cancer, but also prevent autoimmune diseases. So I don't know what other types of information you'd like to hear about our lab.
Yeah, absolutely. So your research is mostly focused on the prevention of lupus, or is there an aspect of treating the disease once someone has a diagnosis as well?
That's a great question. So we do a little bit of both. Most of our studies to date have been focused on prevention as well as understanding biomarkers of disease, but we also have a model that can induce flares. And so for that, we can look at memory responses. So we've been kind of doing a little bit of both primary prevention and disease treatment models in our lab. So from the T-cell perspective, we've been trying to determine what factors are required for the T-cells to cause primary disease and flares. And we've also had funding from the Lupus Research Alliance to study that process, and then funding from the Department of Defense to study flare responses or memory responses in our mice. I will say it's challenging to treat lupus. It's challenging to deplete the autoreactive T cells, at least some of the methods that I tried back when I was a postdoctoral fellow in John Harris's lab, also at UMass. We were studying a different autoimmune disease called vitiligo, which is what Michael Jackson had. And we were able to successfully treat that disease and target that disease because the pathogenesis is very linear, right? Like you can kind of block any step in a pathway and you can help to treat vitiligo. When I tried applying those same types of techniques to lupus, a lot of them have been underwhelming. Like they'll maybe help some disease parameters, but not completely prevent lupus. And I think it's just because of how complex lupus is immunologically.
Yeah, I've heard that, I'm not fully ingrained in the scientific community, but just talking to people who have lupus, people that have loved ones who have lupus, I mean, that shows up not only in the like what you're talking about, the research aspect, but also just how lupus presents itself, how it's found. Like it's just so complex and so nuanced and so individual to the patient that that's why there needs to be more research around it, right?
Absolutely. And there's actually a big push now from the Lupus Research Alliance, who they've funded a lot of our work. I'm very grateful for them. They kind of funded my career transition award, which allowed me to open my own lab, which was really awesome. But they are pushing now for biomarkers to help stratify patients, like to understand, like, okay, you have this type of lupus versus this type of lupus. And so these treatments may work better for patients with these symptoms, and these treatments may work better for patients with these symptoms. They also have a goal of reducing the time to diagnosis, because as I'm sure you know, it takes years for people to get a lupus diagnosis and can be very frustrating for patients. And then there's a lot of trial and error that goes into like which medications you start with. Sometimes it's dictated by how severe the symptoms are that a person has. And other times, like I said, it's just a trial and error. Try this drug, see if it works. Try that drug, see if it works. And I think it's frustrating both for the patients and their families as well as the providers, because there's like no good like gold standard to figure out like, okay, yeah, you have this kind of lupus, this drug class should work. And so we're still kind of trying to figure that out. I think it's like you said, just because it's so different in different people, it presents so differently in different people.
Yeah, absolutely. It's frustrating on all accounts, obviously for the patients, but like you said, the providers as well, because it's a lot of, throwing darts at the wall and seeing what sticks. How did you find yourself in this type of research? Tell me a little bit about like your journey to get here.
Yeah, absolutely. So as I mentioned, I did my postdoctoral fellowship with John Harris studying vitiligo. And actually, that was my second fellowship. I had done a previous fellowship at Mass General Hospital up in the Charlestown Navy Yard, where we were studying different chemokines. So I like to describe chemokines to people as like the signals that tell your white blood cells where to go in your body. And so the analogy I usually give people is if I were to blindfold you and I would go to like the corner of the room and squirt a perfume bottle. Like, how would you come find the perfume, right? You would follow your nose like to the scent where it's strongest, right? So chemokines are like the perfume, right? Like they tell the cells like where to go. And so while I was working at Mass General, Dr. Harris reached out to Dr. Lester and said, hey, we see that those same chemokines that you guys are studying in a vaccine model are really high in our vitiligo patients. And so he wanted to collaborate with us. And at the time I was commuting almost 2 hours one way to get into the city because I was taking the train and then walking from North Station. So I asked if I could actually run the collaboration from UMass. And so that's kind of how I transitioned to my second fellowship. And then when I was getting ready to transition to an instructor and then an assistant professor to run an independent lab, we started talking with Dr. Ann Marshak-Rothstein, who's also at UMass Chan. She's actually a famous B-cell biologist and TLR biologist. That's a type of innate immune sensor. And, we were discussing how the chemokines are also high in lupus. And so we were wondering like, okay, well, are the T cells following the same signals in lupus? Are the B cells following those signals? And that was kind of the project that I mentioned that the Lupus Research Alliance funded. That was my first like big grant to look at the CXCR3 chemokine family in lupus. And so when we were working in vitiligo, if we blocked the receptor on the T cells, like we basically plugged their nose so they can't figure out where to go, we prevented vitiligo. And we actually had a use patent that we generated with Sanofi Genzyme. So it's Genzyme, but now they've been bought out by Sanofi. And so that seemed to work, right? I think there were concerns about actually like trying to get that particular drug into people because they were worried about like off-target effects and things like that. But when I tried that same drug in our lupus model, it like reduced disease and reduced the flare, but it didn't like prevent it, right? They still had cutaneous lupus. That's mostly what I've been studying is skin lupus. And so, I think it just goes to show you that there's so many redundant immune pathways in lupus. And I think it's because the innate immune system plays such a big role, right? Like that whole system is kind of there to prevent, bacterial infections and viral infections and things like that. And it's like sensors and pattern-based recognition. And so you want a lot of redundancy in that. But then if you have these genetic susceptibilities that make it likely that your immune system could start reacting against your own body's tissues, turning it off is more challenging, right? Because you have these like kind of, I don't know, Legos or building blocks is the other analogy I use. It's like you can stick two different parts together and kind of get a similar end result as if you had different parts that you put together, right? And that's kind of how the innate immune sensors work. So so it's definitely challenging and we need to do more research to try to figure it all out. But there are, there's a couple things in the lab that I'm excited about. Like we're starting to also test drugs that are used for other conditions, kind of like off-label, right? Like drug repurposing type studies to block different types of receptors that help the T cells get into the skin because we found a lot of other types of perfume that are being secreted in the skin. And some of them bind to the same receptor, actually. So there's a lot of what they call redundancy in the chemokine system. So different types of chemokines can bind to the same receptor, or one receptor can actually recognize many different types of chemokines. So there's a lot of like complexity in that system. And that's really what we've been studying. And I have like one kind of anecdotal study story I can share with you about. So we had a T cell that we tried knocking out one of its chemokine receptors with the goal of preventing lupus, but we actually made it worse. So then we were like, okay, well, what can we do with that? And so we actually started using those same T cells in a melanoma model, because it's a type of skin cancer. And the mice are surviving better. So it's like, even if you get a negative result, sometimes you can actually repurpose it for something else and actually make something useful out of it. And so that's like one of the like kind of, twisty, windy roads they talk about it in research. It's like usually never a straight path to finding a treatment or a medication. Sometimes it's serendipitous, like that example I just gave you, which we're hoping to eventually move forward. Or like another like old school example is penicillin, right? Like that was kind of like an observation that was made in the lab, just because, like they were growing this culture and everything died because it had this mold in it. And then you can use make the drug from the mold and treat bacterial infections in people. So I think it's, being curious, being open-minded, and like, when you see a result and if it's not what you expected, like thinking about it and thinking, well, what utility can I get from this result, even if it's not what I expected? So
yeah, absolutely. Yeah, that's crazy. Like just the different like adaptations to these different treatments, and like yeah, it just really, this is like so outside of my wheelhouse as a marketer, right? Because it's like I'm very like what is it, like right brained, like I'm like more in the creative land, and like this is such like a process. I used to work with a clinical research facility and do marketing for them, but yeah, it's just so fascinating for me to hear sort of the back end of the process. So I guess what stage is your research in? You talked about mice a little bit, so it's not in like the clinical research phase at this point.
So I do mouse, dog, and human research in my lab. So for the mice, we do use preclinical mice for testing. And, we do try to reduce the number of animals. We use, there's, the 3 R's in research: reduce, repurpose, reuse data, right? So that you don't use mice unnecessarily. So we do try to abide by that. But I haven't really found a better way to do some of the preclinical testing, because cells in a dish don't necessarily behave the same way like a physiological being does, right? Like, because you have to have your kidneys that can, metabolize stuff and secrete it in the urine, right? And but one of the ways that we're trying to actually like help animals is through our dog studies. So for the dog studies, we collaborate with veterinarians at Tufts Vet School, because pet dogs can develop lupus spontaneously. Specifically, discoid lupus is pretty common in like German Shepherd breeds and their crosses. And so we have like basically, they come in to get a diagnosis just like a person would. They get a skin biopsy taken, and then there's tissue left in that block after the pathologist reads out the tissue to give them the diagnosis. And so I reached out to the veterinary pathologist and I said, can we use some of the tissue that's left over? And he said, sure. So we actually did some gene expression studies on dogs, pet dogs that spontaneously developed lupus. And now we're starting to like actually try to treat them. And so one of the most exciting emails that I got, we published that study describing all of the gene expression in the skin in dog discoid lupus. And I got an email from a vet in the UK who quoted the last line of the paper, said, "I loved your paper, especially this line where you were hypothesizing that this type of drug might work." And we went ahead and we tested it and it worked. And now we're publishing a case series, of like 7 dogs that responded to Oclicit, and it was the name of the drug. So that was really rewarding, right? It's like something that we found, and then the clinical people ran with it, and it's actually working. And now they're using that as a mainstay for treatment of cutaneous lupus in dogs, as well as other skin diseases actually in dogs. It's just a kind of a general anti-inflammatory drug. And I think people are trying to find more targeted ways to target like specific molecules in that pathway, so it's not quite as broad spectrum. I mean, that's always the goal, right? You don't want to like completely immunosuppress a person or their pet.
Right.
So yeah, and then and there are some case studies in humans, because there is a human drug equivalent for that drug. But a lot of the drugs that end up in people, they have to go through animal testing, and some of them are developed in parallel, like with veterinary medicine. So they actually can be used for treating animals, companion animals. And, they do have drugs also for like farm animals and things like that. But
That's
yeah,
so
and then
cool. You like always hear about like the horror stories of animal testing, but you never think about that it could actually like benefit the animals in some way. And especially, I mean, like who doesn't love dogs? Like, yeah, what a like cool. Yeah, I can imagine like getting that email and being like, oh my gosh, like something that we worked on like here in Worcester is affecting dogs in the UK. Like that's crazy.
yeah, it was really exciting. And we do work with human samples as well. We do different types of studies, right? We've gotten blister biopsies in the past. So we have this machine, it has like a little metal like canister, and you can like put it on your arm or your back or wherever you have a skin rash. And it provides like a little bit of negative pressure, so you get like a blister that forms. It's like a tiny blister, just like I've gotten them before on my heels. I used to run cross country in high school, so I used to get blisters all the time. So it's like that. And then we can aspirate the fluid out, and we can analyze the fluid part of the skin and then the immune cells that got into the skin. And there's benefits and downsides to that, right? Like there's minimal processing, right? We don't have to like break up the skin tissue, because the way that we usually do that is with an enzyme, and that will like chew up the tissue. And sometimes that can, chew up markers on the surface of a cell. So we don't have to do that, right? If we're just pulling it out of the fluid, you can just put the cells right into, it's a machine called a flow cytometer that we use to like look at the different immune cells. But the downside is you're really only getting the stuff that's like near the top of the skin. And particularly in like discoid lupus, there's stuff going on deeper down in the tissue. And so for those studies, again, I collaborated with a pathologist at UMass, Dr. April Dang. She had old discoid lupus biopsies from people. And so we collaborated with the scope core at UMass Chan, it's our microscopy core, to do what they call spatial transcriptomics. And so that, I don't really know how to describe this well. So there, you basically get a tissue section from that little biopsy, and you put it on a slide, and you put it on a microscope that's attached to this special machine, and then the computer. And you can actually like basically draw, like I draw like a polygon shape or whatever around the parts of the skin that I want to cut out. And then the machine will shine a laser on that part of the skin, and it has like a fluidics chamber. So like it runs fluid over it and collects it into a well. And then we can do sequencing on that. So you can literally take different parts of the tissue, that's why they call it spatial, and know what gene expression is happening where. And we also have different types of stains. We can do up to 3 stains to figure out like what, like for me, I stain for T cells, right? And I pull the T cells out from like the deep dermis versus the superficial part of the skin. And I say like, what's different? Like, why are they hanging out down there versus up here, and what's going on in the tissue? And so we have a preprint describing that. We're trying to get that published in a scientific journal. So yeah, I think we try to do mouse, dog, human, right? We try to minimize the number of mice, but we kind of have to do some of the testing in the mice because we don't want to like hurt someone's pet dog or hurt a person, right? Like again, I haven't really found a better way to do that yet. But then we're minimizing, because if we find something that's already a drug class that, like we did with oclacitinib, that will work in pets, and then someone's willing to test it, right? That's the other aspect of all of this. Like you have to have someone who's willing to participate, and participation in clinical trials is like so huge and so important, right? It's like you're doing something bigger than for yourself, right? You're helping all the future patients. So that kind of thing I think is also very important. But yeah, and then if it works in the dogs, then you can jump to people a lot faster, right? Like you can skip some of the other like vertebrate animal testing that needs to be done. And so that's kind of the model that I've adapted for my lab to try to like, help people and pets and minimize the number of animals we use expressly for research purposes until we can get a better handle on lupus. So.
Yeah, absolutely. That's great. Tell me a little bit about your interactions with the lupus community. If you have any, like, I mean, no, you know folks through the foundation, but what does that look like? Like, what sorts of conversations are you having? Like, how does it inform or inspire your daily work? Talk a little bit about that.
Absolutely. So I found the Lupus chat folks on back when it was Twitter. Now we've all migrated over to Blue Sky. And they're just so fantastic. So it's a team of 4 women who run the Lupus Chat nonprofit organization. And it's literally just that, like on a Sunday afternoon, I think it's 3 p.m. Eastern Standard Time. They'll have usually a set of 5 questions and they'll answer from their perspectives, but they'll also encourage patients to jump in and answer from their perspectives. And so I can't remember how I first came across them. I think I was just searching for like lupus support groups and I found them. And so I started by just listening, right? And just listening to their stories, listening to the community, like what people are experiencing, what was going on. And that was just really powerful just to like hear what's most pressing to them, right? Photosensitivity definitely is annoying, but one of the big things that people complained about was like brain fog. And so we've actually started moving into studying other organs, including the nervous system in lupus because of like, you know, a drive to wanting to help people with what's bothering them most, right? And then just, you know, having me there, they welcomed me into the community and they even like featured me on Why Research Matters during Lupus Awareness Month in May, a couple of years ago, I think it was. And so I just talked about, you know, what we do and the perspective and like how important it is like for us to get some human samples, like people who are willing to come in and give blister biopsies. That was huge, right? Because we could tell like what was actually recapitulated in dog and mouse that we could say, okay, this may also work for people or this may not work for people, right? That's equally important. And so just talking a little bit about, you know, why we do what we do, how we do what we do. And it's just been really great that they welcomed us. And then obviously I know Beverly, I've known Beverly for years now. And we actually, the first social media liaison was one of my former students from Clark. I adjuncted at Clark and she did social media liaison work. And so she worked with Beverly. So I can't remember how that connection worked, but I think she was already working with Beverly. So she connected me with her. And so we had this really nice connection from the beginning. And so that was kind of when we started doing Lupus Day at UMass in conjunction with Lupus Foundation of New England. And that was originally designed to be, we had like a scientific portion and a patient portion so that for the researchers, you know, we would get some scientists to come and talk about the research and talk about cutting edge, like what's going on in the lupus field. And then later in the day, we would have like a physician panel so that the patients could ask questions like, you know, what does this mean? What are the current clinical trials that I could participate in, right? Things like that. And we encourage people to attend both sessions, right? We want the researchers to understand like the patient perspectives and like what's bothering them. But we also want the patients to hear a little bit about the science that's going on so that they can kind of make more informed decisions. And I think science communication is also like really important, like trying to explain stuff so that people can understand it. And some of my medical students actually have worked on that with me. We have 2 articles specific for lupus. One was by my student, Haya Raif, who's now doing a dermatology residency. She wrote about what is photosensitivity, basically, can kids be allergic to the sun is the title of her article. And so she talks about lupus photosensitivity as well as a couple other conditions that can cause sun sensitivity. And then my student, Warda Abdi, actually interviewed one of the Lupus Chat ladies. It's a mother-daughter team. So she interviewed Maya and her mother for the What is Lupus science communication article. And so I think, you know, trying to incorporate all of that is also really important. And again, I'm just really thankful that, you know, we have all these teams of amazing people who are willing to like listen to me and, you know, all my nerdy science talk, I guess.
Yeah, I mean, it's really, I mean, it's extremely important. And I think for patients too, that sense of like hope and knowledge that someone's out there like trying to do something about this, about this condition that they're living with on a daily basis, I think is really important. And knowing like the faces behind the research and you know, hearing folks' perspective is just I think can really change someone's attitude and be a bright spot in their day. So yeah, I guess to wrap up, what would you say to people that are kind of making the financial decisions about where the money goes, if it goes towards lupus research, and also if there's anything that you want to share with current patients as well, but mostly, I guess, policymakers or sort of financial decision makers.
Yeah, well, I mean, as you know, we're kind of in an administration right now that is cutting a lot of science and science infrastructure at the federal level. And I will say that science has long had bipartisan support. So without me picking sides, there've been past Republican presidents, past Democratic presidents who have supported the NIH and its mission and the federal budget to do as such. It's also a relatively small fraction of our overall federal budget. And so I think any cost cutting that they're trying to do now is not actually going to be effective. I think there's some statistics that every dollar invested in NIH research yields $2 to $3 in return. It also results in job creation. I mean, UMass is one of the largest employers, or maybe is the largest employer in Worcester County. And, when people talk about overhead for research, I think that's one of the things that's been kind of thrown about a lot in the news lately. So the way it works for NIH is, for every grant you get, for every $100,000 grant, this school will also receive about $67,000. So it's not that they're taking that away from the grant money. It's that the NIH would pay $167,000. $100,000 of that would go to our lab, right, which covers my postdoc salary, the animal techs that help take care of the mice, the reagents that we use to measure everything, all those types of things. And then the $67,000 would go to pay our administrator, my wonderful EBS personnel, Marta, who helps, keep our floor clean, the electricity, the plumbing, right? So there is definitely a cost to research, but again, it results in job creation. It helps make medicines. I mean, we have many options nowadays. I mean, cancer used to be a death sentence, right? And nowadays people are getting functional cures with the CAR T-cell therapies, for example. Lupus used to be a death sentence, right? And now people can live relatively normal lifespans if they're maintained on Plaquenil or whatever drug they need to keep their lupus in check, right? So I think that it's a little short-sighted, and it's very short-sighted. And I would encourage people to, lobby for science. I actually attended the March for Science in Boston this past Friday with my friend who's a professor at Tufts. And, I feel like it's powerful, right? Like the civil disobedience and the peaceful protesting, we need more of that nowadays. But in terms of your other question about people deciding whether or not they want to give to research, I mean, the Lupus Research Alliance is a wonderful organization. They've done a lot for lupus and for lupus researchers, like myself included. I mentioned that they gave me my first big independent grant to be able to open my own lab. I think they do a fantastic job. They do a lot with education, advocacy, outreach. They have a Capitol Hill Day where they go and they lobby the lawmakers to understand what lupus is and why we need science funding to help find cures for lupus. And so if people are, deciding whether or not they want to give to, any of the organizations I've mentioned, like Lupus Research Alliance, Lupus Foundation New England, Lupus Chat, right? These are all nonprofit organizations. You can get tax deductible donations, and you can know that, especially with LRA, they're very transparent about where all of their money goes. They use majority of it for funding research. I think they use very little of it for advertisement and overhead. I think you can check, they have the charity navigator, right? You can actually look them up and see where all your stuff is going, all your money is going. Yeah. And there's other ways you can participate as well, like non-monetary ways, right? You can do the lupus walks that are hosted by LFNE or LRA. You can go on social media, join the support groups, be vocal, be supportive, help educate people for what it is. I think there's also maybe less so now, but still misconceptions about what chronic illness and disability does, right? People, they're not lazy. They actually have a condition, right, that sometimes can make you bedridden. And so I think helping people understand that, and you know, helping people advocate to find creative ways to find work and, you know, community and belonging, right? And I think that was another nice thing about Lupus Chat is like, sometimes the patients are saying like, you know, oh, I'm joining today from my bed, right? Or I'm joining today from, you know, my infusion at the hospital, right? So I think, you know, now that we do have technology, right, there's ways that we can be, you know, even more inclusive of people with all different abilities and talents. So, yeah.
Yeah, absolutely. I love all of that, and I agree with everything you said. It was a big weekend for all different types of marches.
Yes.
So, thank you so much, Jillian, for taking the time out of your day to chat with us, chat with me about all this. I think just giving like a behind the scenes look into what lupus research looks like, and like I said, seeing the faces, hearing the voices of the folks that are on the ground doing this work every day, I think just goes a long way, not only for education, but also like motivation, like life, you know, just being more hopeful about the future of lupus and symptom management and all of that. So thank you for the work that you do, and thank you for today. Do you have any places to send people if they want to learn more about your lab, or if they're just interested in reaching out or something?
Yeah, absolutely. I have a lab website. We have all our science communication articles is like one of the tabs. And you can see all the current personnel, students, everything, and the papers that we are working on, like the deliverables from all of our research funding. And you can, you know, reach out to me via email, and I'm happy to chat.
Great. I will put the link to that in the description of this video. Thank you again.
Thank you
And,
so much, Jillian.
yeah.